welcome all participants all over the world. I am very happy that SimCell is organizing such educational events where we can share new pathways of microcalorimetry in medicine and related disciplines. I was using this method for research purposes since more than 20 years. And in the last 10 years, we are going into the clinic and see how this method can detect infection and go even one step further, identify the pathogen, and even one step further, identify the antimicrobial susceptibility. This became much more important in the last years because of the emerging of resistance, and I will speak both from the aspect of a clinician who is doing also translational research. Why infections are important, I would like to emphasize on this slide. On the right, you can see our body, human body, also called Homo sapiens, the wise man in Latin. And we have about 10 to the 13 our own cells. If we count the bacteria and fungi which are on us and in us, then we see that there are 10 times more bacterial and fungal cells. So we are only 10% human and we are 90% from microbes. And therefore it's also not surprising that we are full of bacteria on the skin and mucosa. Here, a normal skin flora has 100,000 bacteria per square centimeter. And whenever the skin is damaged or a mucosa is inflamed, then bacteria can translocate and enter our body. Bacteria are much longer on the world. If we see this scale of the evolution and the Earth, which developed 4.6 billion years ago, then the first life forms indeed are microorganisms, 3.5 billion years ago. And then we have biofilms, and only at the end of this evolution spiral is us. So we see how much bacteria are more adapted to this planet than us. If we put the scale since life is on this planet into 1 year, then the first inhabitants were bacteria coming on 1st of January of this year on the planet, fungi about half a year later, and mammals only on the 24th of December, and our species only the last 4 minutes of this year. So we see that we are in a symbiosis, bacteria as the microorganisms and us as the macroorganisms. And antibiotics, we are using only the first 5 seconds against bacteria. So the bacteria will always be smarter than us. And if we implant also something foreign in our body, which we are using commonly in medicine, the implants, then bacteria in addition form also the biofilm, which becomes an additional problem because they are going onto the surface in a sleeping phase, and we have difficulties to detect them and also difficult to treat them. But this second challenge, so besides identification and resistance, which is the biofilm, we need to face because, as you see and know, that modern medicine is using different implants to improve quality of life. If we look now on the big scale, then we see that different implants are implanted worldwide, but the infection rate is about 5 to 10%. So every 20th or every 10th of our patients will get an infection as a biofilm. So we need efficient methods to diagnose these infections, which is the first stage to treat them efficiently. And I will speak in the second half of my talk about calorimetry, which is a very simple method to detect viable cells just by detecting their heat of metabolism. Here is a nice electron microscopy picture of many bacteria which are growing on the surface and producing an infection. So if you want to achieve high cure rate, the first task is to diagnose these microorganisms reliably, because then we can adapt antibiotics and often also surgery. The first task to have diagnosis for example, when we look for joints, both native and implant joints, is of course the joint aspiration. So we have a fluid in the joint which we can aspirate and send for culture. There are also molecular tests available, but the problem is that most bacteria, as you can see here in red, they are on the implant itself. So we often use inflammation markers such as leukocytes counts or pathology or biomarkers. But if we have a more sensitive method, which can detect already a couple of bacteria, which will not be detected by culture or molecular tests, but will be detected by their heat, then we have much improved our diagnosis. The same is true from samples which we can get during surgery. You can see here what we usually do. We take several tissue samples. We send them also for histology. And sometimes we do also sonication. If there is an implant, here as an example is a knee prosthesis, then we can send it to sonication. You can see a sonicator here on the right side, where we dislodge the bacteria which grow as a biofilm on the surface. Usually we use 40 kilohertz of sonication, and then these bacteria which are in the fluid, we call it sonication fluid, can be detected either in a classical culture, but even better in biocalorimetry, because as we will see later, calorimetry is much faster than the conventional culture. These cultures shown here are usually after 3, 4, 5 days, but it can take up to 14 days that these colonies can be visibly detected and then further identified. So the advantage using not a visible growth, but their metabolic activity, the heat, is that we are much faster before we see these colonies. Each of these colonies, you can see on the left side, for example, from tissue, has millions of bacteria. So it's quite late that we can say that this culture is positive. So the new diagnostic options are then were investigated. I mentioned before molecular tests. These were very attractive because molecular tests, which detects the genome of bacteria, can detect also dead bacteria, those who are not growing. However, molecular tests have disadvantage that they are also not very sensitive, but also not very specific, because we have commonly contamination of a DNA, either during sampling from the patient or the transport to the lab or in the lab itself, because most of the reagents for PCR or sequencing are produced from bacteria and can also then detect false DNA, which was not present in the patient. So logically then is that we look for something which is produced by living cells, especially if they are replicating, and that is their heat. Asking now how much heat is produced by our cells, we can see here some normal cells, fibroblasts, adipocytes. They produce about 40 to 50 picowatts per cell. Granulocytes, lymphocytes, so those in blood are producing less heat, but there are so much of them that we can detect them as well. The most advantages are bacteria, because as you can see here, they're replicating. Typically every 30 to 40 minutes, they are double as much bacteria as before. So we have an exponential increase of heat production, as seen here in a typical heat flow curve, where we have at the beginning some time that the bacteria are not yet in a growing phase, but then they go into the exponential growth. And parallel to this, we can see the heat flow, not in picowatt, but in microwatt. And if there is a lot of bacteria in good growing conditions, then we can have much very hot. And the better we can detect the heat, the shorter is the time to detection. I will show you some applications and results we have done originally with the thumb instrument from TA Instruments, the only available instruments 20 years ago. Now we have much more sensitive and easier and automated instruments which are tabletop and have exactly the same or better performance. For example, here we put two important bacteria in medicine. Staph aureus is the most common cause of infections, and for implant infection is Staphylococcus epidermidis. If we use a concentration which is quite low, 10,000 colony forming units per milliliter, just as for comparison, if we have an acute infection, we have 10 to the 8 or 10 to the 9 bacteria, so much more. But if we have this concentration, then within hours we will already, or minutes already, we can see an increased heat production. These are now duplicates, and you can see also how repeatable this investigation is. We went then by diluting this bacteria from 1 million, so 10 to the 6, down to 1 CFU, so only one bacteria, and we see that we also detected it, and the curve was quite similar, just it took a little more time, but still it was only a couple of hours to detect them. The one sample which theoretically had less than one bacteria, so statistically 0.1 cell, was negative because there were indeed no bacteria inside. So having these encouraging results, then the question is, of course, where we can use it. And I will mention some applications, saying that none of those is now currently used because there was no instrument which is easy to use. But knowing the results from the past decades and having a better technology, we can maybe rethink to use it. I will start with platelets. Platelets are used to transfuse in patients who have coagulation disorders and they are bleeding. So we need to transfuse these platelets, but the problem is that their shelf life is only 5 days. So the blood banks really need always fresh platelets, which needs immediately to be transmitted to the patients. Because infectious risk is increasing after day 5 a lot. So if there would be a screening tool, which we don't use it now, then we could easily use them longer, the platelets, or increase the patient safety. Because you can imagine if you infuse not only platelets, but also bacteria which are contaminating these platelets, then patient will go into septic shock and can die. Therefore, we tested this and we inoculated different bacteria, Staphylococci, Streptococci, and even gram-negative, Escherichia coli. And you can see that in all bacteria, we were able to detect up to 1 or at least 10 bacteria within several hours. This was published in Transfusion already in 2007, but was not used because, as I said, there was no instrument. But when we use the 10 to the 5th, so higher concentration of pathogen, which is the most dangerous for the patient, then between 1.8 hours and 3.2 hours, we were able to detect the most dangerous bacteria, Staphylococci, Streptococci, and Escherichia coli in contaminated platelets. So if it works for platelets, it should work also for blood. Now, what we use is we take blood samples and put them in the blood culture machine, which detects positive cultures. You can see here on the left picture, a positive one, by changing the color, which is an indicator that there is a CO2 production. So very insensitive method. We have a CO2 sensor and a colorimetric, so change of color, which tells us if the blood is containing bacteria or not. So its limitation, it takes several days to have positive results, and therefore we tested maybe we can use blood in the colorimetry. So we did in parallel use a commercial blood culture system, in this case BactAlert. Today they are also others, And instead of 7.5 milliliters, which we put in the blood culture bottle, we use only 1 milliliter for the colorimetry. And here are the results. So 7 milliliters of blood compared to only 1 milliliter in colorimeter was detecting all pathogens much faster, especially those who are very virulent. Staph aureus, instead of 12 hours, we were already at 4 hours positive. So gain almost 8 hours. And this is very important because each minute is counting to start an appropriate antibiotic treatment. There was one pathogen, though, which was detected later, and that was Candida albicans. And another one, but in most cases we were faster and therefore more sensitive. There can be some optimizations done by adding detergents and anticoagulants, and this one is still in process. But what we have more now is data on synovial fluid. I showed at the beginning that we can puncture the joints to see if they are infected or not. And what we do today is mostly counting the leukocyte count, which is not very distinguishing if this is degenerative, inflammatory, or infectious, septic. The reason why these methods, counting leukocytes, is not so sensitive and specific is that the leukocytes are very active. You can see here in this movie how granulocytes in the joint is going after bacteria and trying to eat them. So this induces a lot of inflammation, but we don't know if by counting the leukocyte, if this is really induced by infection, such as in this case, or it is induced by other inflammation. One of those is degenerative disease, osteoarthritis. The others is crystal joint disease, where the crystals are inducing or are indeed bacteria. So we tested it in the colorimeter, and you can see here that there was always a heat, but then because these were only leukocytes inside without bacteria, these leukocytes produced less and less heat because the leukocytes died. So it was never an increase of heat production. And interestingly, the same was in crystal induced. So it's called also gout or pseudo gout. We have a lot of leukocytes. They produce up to 50 microwatts of heat, but after a couple of minutes, these leukocytes were not dividing. So no bacteria inside, we just have a reduction of heat. However, in arthritis, we see exactly the opposite. So the leukocytes here, you see a peak, but then 1 hour later, the bacteria start producing heat. And we have now 2 patients as an example. Both have a streptococcal infection. You can see how heat is producing and exponentially after 4 hours. So we have a rapid test which can distinguish if this is inflammation caused by infection or by other pathology. Therefore, logically, the next question is, okay, if this works for joints, maybe this works also for cerebrospinal fluid, which is even more important because meningitis, bacterial meningitis, is a medical emergency. So we didn't go to patients to test it, but we went to an animal model, a rat model, where different bacteria can be injected, pneumococci, meningococci, and listeria, to induce the meningitis. And as a control, we inactivated these bacteria by heat, so they were dead, but we injected them as well. And look, here are the results. So if we use 10 microliters of cerebrospinal fluid, we were positive within 2 or 3 hours. If we use even less fluid, down to 0.1 microliter, so it's 100 of nanoliter, it was also positive just a little later. So we have a very fast, rapid tool, which is also very specific to detect bacteria in cerebrospinal fluid within 2 hours. And that is no other test which is so fast and so reliable. So then we ask as a last question, can we go to those pathogens which are growing very slow? For example, Plasmodia. Malaria is actually the most common infectious diseases, killing more than 1 million people still every year, mostly children. So we used blood and tested it in the colorimeter, either the uninfected erythrocytes or infected by Plasmodium falciparum, the most dangerous malaria form. And indeed, we see we have also here the positive heat. And we could see that even at low parasitemia, 0.5%, there was heat detection. And then we went with by dilution of the blood down to 0.008% infected erythrocytes, and that was still possible to detect. So we can summarize that no matter if you use highly virulent, faster replicating bacteria or even parasites, we can detect them. And maybe we can use this method also to test new antibiotics, new drugs, to see how quickly they can kill bacteria, because efficient antibiotics will prevent development of heat. So I will now go to a clinical study, which we have done in cooperation with Italy. On one side, that was infectious disease person Martina Maritati and my PhD student Sarah Zung, who tested 22 samples from synovial fluid. They were actually frozen after they were collected, and they were transferred to Berlin. We see here which were the pathogens detected in normal culture, and they were tested in the calorimetry. And we see that all the positive ones which grown in culture were also positive in the calorimetry. Despite this freezing process, there were some which were false negative, and those we will now test how we can, if they were really negative or they died during the transportation. So but despite of this process, we see a consistency of more than 80% between the cultures which was done there in Italy and the calorimetry which was done here after this freezing process. If we exclude those who were also now negative, the consistency even increase. And also here, the important advantage is the speed. So we were positive very quickly within the first day. Finally, I would like to thank my research group, which is working on these experiments, but also the clinical team, which collects all the samples, and would like to finish with motivation to continue research, both in the preclinical setting and the clinical setting, to make this step forward. And my dream would be to have not only early detection, but also identification by using the data bank. And we see that there is a high consistency with typical calorimetric curves, and also by early detection of antimicrobial susceptibility by adding antibiotics already in the calorimeter. And if there is no heat production, then we can say that these bacteria are susceptible. Thank you very much.
Thank you very much indeed, Andrej. That was super, really interesting insight into how to use this in some really important clinical examples there. So thank you very much. To our audience, I'd like to say thank you very much for listening. I know some of you had to go, but if you have any follow-up or questions, please feel free to send me an email via the email which you got for the registration here. I'll follow up with you and I'll send you the recording as well. So don't worry about that. But For those of you who can stay, I know some of you had to leave. I'd love to hear your questions now, your thoughts. If you'd like to have any discussion now with Andrej, please drop your questions into the Q&A or into the webinar chat, and we will get straight to it. So I have a question for you here already, Andrej. So basically in chronic infections, first part of the question is, would you expect the bacteria there to be biofilm in chronic infections? And if it is biofilm, is there enough metabolic activity to be able to detect it, because they're often metabolically inactive or dormant there?
I see this question from Casper, and it's actually a very good question, because the biofilm, the most typical part is that they are metabolically in a low active phase. And that makes it so difficult to detect. For that reason, I presented sonication. So if we first remove the bacteria, so we planktonize them, we wake them up, then they're growing. And this is what we do today, not only by sonication, but also when we collect the tissue samples, we're actually looking for planktonic bacteria. And the colorimetry would do the same. So either we use tissue, then we have the planktonic bacteria already, or if it's implant, I would do sonication first, and then they are growing. What is important, and therefore I like this question, is that if bacteria are not replicating, they will not produce much heat. They will be like leukocytes, probably even going down. So we need to bring them to be metabolically active to grow. And there is not much growth needed, but it's just that we see an increase. And with artificial intelligence, probably we can detect it very early when the curve goes down and when it goes up. And that will be then the early diagnostic procedure. That this works, we see with plasmodia. So these parasites which are infecting the erythrocytes, and where they are replicating very slow. They need, I think, 20 days. So, but even then we can detect them. I put at the beginning with a question mark also tumor cells, because tumor cells are also replicating faster than our own cells, but not as fast as bacteria. So first we should focus maybe on bacteria, but then I can imagine colorimetry as a good tool also for leukemia, for example, and also to test the chemotherapeutics, if they can, how they can kill tumor cells. So it is, I think, a long journey we still can go.
Great, and super broad applications. It feels like anything you can really think of, you can almost imagine an application for biocalorimetry there. So it's really interesting. Now, I have a follow-up question here. Do you think it could be used, so biocalorimetry used for fast diagnosis of hard to grow neuroinfections like borreliosis or syphilis in spinal fluid, because they tend to be quite low concentration? So yeah, what's your feeling on that?
Interesting. I actually didn't show that slide. Now I see the question came. Those who bacteria who belongs to spirochetes, Borrelia or Treponema, which causing syphilis, they are very easily to detect, because not because of replication, but because they produce so much heat, because they're turning around. They are like drilling into the tissue, and therefore we can detect them very easily. Borrelia and Treponema are actually not growing in conventional cultures. We use animal models to cultivate them, but by colorimetry we detect the heat of the metabolism. Yes, I think it would be another good tool. In those two diseases which was mentioned also by Casper, though, there are also other methods, serologic methods, which can be used, or PCR methods. But the colorimetry is the only one where we can see the effect of drugs. And I would add to those two diseases also tuberculosis. Mycobacterium tuberculosis is very slow growing, sometimes for weeks, and colorimetry is able to detect them and also do the susceptibility within 1 day.
Fascinating. we have another question here. So is every bacterial species, does it have a different kind of heat trace? And if so, you know, how do you use those? Is that what's used for identification? You know, how unique are the values coming from each bacteria?
I like the word heat trace. We call it thermogram. And we can say if there is a lot of heat, for example, 500, 600, or 700 microwatts, that's probably a gram negative, especially E. coli. If it's, let's say, more about 40 to 50 microwatts, then it's Staph aureus. If it's only 10 microwatts, then it's probably an anaerobe. And we have published an article where we tested all anaerobes to see which growth medium is the best. And it was thioglycolate, which allows a lot of heat production, which means a lot of replication. So we can use it also for the fastidious organisms, which needs either absence of oxygen or presence of higher CO2 gas, so that they can better grow. So I'm sure we can improve also those detection and susceptibility testing. Now, to do the antibiogram, so the susceptibility testing of anaerobes, it's very cumbersome because we need a special chamber, and usually many days before we can say if it's susceptible or not. In the colorimetry, it's in 1 day.
Wow. Fantastic. Can I just have a look here? If you have any more questions, please drop them in. I have a couple of my own, so I'll squeeze myself in now if that's possible. But you talked about kind of, you know, the collaboration you had in Italy and the kind of the bank of kind of thermograms you have, and also a little bit about AI. So this is related to the actual identification, right? How do you see, is that something that can be done now? Is there enough data collected, or is that something that can be done in the future? And if so, what's required to have, you know, really good identification from this technique, do you think?
Everything what I mentioned now is, of course, off-label, because we need to show in clinical studies the correlation. Whenever you have a method which is more sensitive, then it's difficult to compare with the standard one, because whatever you find, you need to prove also that it's real and not a contamination. So clinical studies are needed, and lack of this clinical studies is the main barrier. Why is it not widespread clinically used? But I must say there were also no instruments available. This machine, the TAM machine, was very expensive and a lot of handworks. So by doing automatization and easy to use small instruments, I'm sure that this will be part of routine lab. MALDI-TOF is another method, so mass spectrometry, which was not used 12, 13 years ago. Now is a part of every laboratory diagnostic.
Nice. That's a very exciting thought, Andrej. You know, is this where you think it would go to eventually, kind of being on that level where it's kind of routinely used in the clinical labs? Great. So how about the discriminatory power of biocalorimetry when you have multiple species in a sample? If you had a clinical sample, do you see that? Can you differentiate them? How does it look when you have a multi-species infection?
Yeah, I think this is now the challenge. First, you have different isolates which are in different metabolic states and also in different numbers. And then if you have even mixed infections, so you have 2, 3 or more pathogens, then you have, of course, the heat is a sum of each of them. So just by looking at them, it's impossible to identify. So we need this pattern, and also artificial intelligence was not available 10 or 20 years ago. So this technology on the software side can tell us. We can see the same development also in the sequencing. Before, sequencing was very expensive, very long. Now sequencing by having a good software can help us identify also mixed infections very quick. So the same development will be needed also in colorimetry, to have a large data bank, and that will then determine when this become a routine test.
So we need to keep using it, keep depositing the data, and the more data we deposit, the more powerful the technique will become, I suppose.
And then, if I can add something, in comparison with molecular methods, which are now so successful for those difficult to grow pathogens, it cannot distinguish if these bacteria are dead or alive. Whereas colorimetry will always detect only live bacteria. So we can test the efficacy of the treatment. If the DNA is there, but there is no heat, we know that the bacteria are dead.
Of course, yeah, very interesting. I have a question about the barrier to widespread clinical use. So you've never had a barrier to this. You've always, right from the very beginning, even if the instruments were difficult to use and cumbersome, you've always used it. So from your perspective now, what are, I guess the barriers are falling, but are there any barriers left to widespread clinical use? Is it what we've already talked about the databases, or is there anything else that's kind of standing in the way of this being in every clinical lab in the world?
Beside the database and the software and of course the machine, I guess if I can have another dream today is allowed to have dreams, is to have a perfect, just a kit where everything is already inside. So you don't need to think which medium to use, so you just have a sample and you put it in and you put it in the machine, and the machine tells you an hour or two later, no, not only that there is bacteria inside, but also which bacteria and which susceptibility. And this test kit needs to be developed, and I think this will be the direction now to test which growth media, which antibiotics should be given in. And now I come maybe to the most difficult part of the development. We need to standardize it then. Currently, there are microbiological standards in USA, the calSI. In Europe, it's UCAST. So there are two established cutoffs, what is resistant and what is susceptible, or what is intermediate susceptible, and we just need to repeat the same with the colorimetry. So we will have a new parameters which will be not called MIC, so the minimal inhibitory concentration. I guess it will be called MHIC, minimal heat inhibitory concentration. And that will be maybe the next parameter which will be used then for treatment.
Interesting. That maybe you've already answered this question now. Actually, I've got a question here from Christina. So in diagnostic setting, how would the UCAST breakpoints be implemented? Is this how you imagine it will be done?
Yeah, I think we will definitely start with the established cutoffs, so the UCAST MIC breakpoints, and we will see how they correlate with the heat. But they will need to be adapted, because the EUCASTs, for example, they say you need to look for growth after 24 hours. So any susceptibility in UCAST is at least 24 hours. Colorimetry, as we saw, is within a couple of hours, 1 to 3 hours. So we will need to adapt it, and there will be different values which will tell us if it's susceptible or not. And that will need a lot of help, and maybe a new committee will need to be established to get through this cumbersome work. But with software, I think we can be very quick, and definitely EUCAST or calSI breakpoints will be used as a comparison.
Fantastic. Question here: how may we access your tests in the UK? We have a fantastic distributor there in the UK, Mero Scientific, and I'll put you in touch with them. So that's no problem there. Another question: so depending on the medium or liquid you put your clinical sample in, how does that affect identification, and also how does that affect identification with polymicrobial samples? I guess that's kind of the trade-off, depending on the media you put in, depending on the bacteria go in, how does that affect the differentiation power of the thermograms.
Yes, the thermogram is dependent on the medium, that is true, and of course of the pathogen, but we can add also a third player, that is the antibiotic. And I think in combination, different media, different antibiotics in different concentrations and different pathogen will lead to the proper identification and in the same time already susceptibility testing. So maybe that is one of the most promising advantages of this method, that you don't need to first do the identification and then the susceptibility testing. You can do it actually in the same run, and detection as well. So not now is first detection, then identification, then susceptibility testing. Here you can do with the proper kit now, I think all at once, and therefore within hours, hopefully get all of them. Which medium to use? Yeah, it will be a combination, because we saw, for example, Candida or needs a special medium. It's a subaero medium. Anaerobes need another one. And based already on media, we can see if it's more growing anaerobic, then it's probably anaerobic pathogen. If it's only growing in subaero, then it must be more likely a fungus. And we can also use selective media, allowing only growing of gram-positive or only gram-negative. So it will be a play of these different parameters.
Yeah, interesting. I think that's often a light bulb moment for people when you you understand the flexibility that you have with culture-based methods, all the different media you can use, all the different indicators, they're all still there with biocalorimetry. You can use them just the same. It's just that the sensitivity and the time to detection is a lot better with biocalorimetry. So it's really interesting.
Then if I can also comment on that, is a good point you made. We all went from culture-based to the non-culture-based molecular methods because of the advantages we expected. And now it seems by having better technology, we're going back to culture-based method, not for morphology, but for metabolics. And colorimetry is more sensitive than for what it was originally developed. Originally, it was developed to see the degradation of substances such as drugs or polyethylene. So having now bacteria, we see a lot of heat. So from somebody who is doing the whole life colorimetry, microorganism world is very hot.
Nice. Let's have a look. We still have questions coming in. What are the major interfering substances from Christina? I'm not sure how to interpret that, Andrej. Do you know? Yeah, interfering substances. Christina, perhaps you could clarify that a little bit. Do you know what Christina means there, Andrej?
While Christina is maybe clarifying this, I can add another advantage of colorimetry, and that is we give patients often not only one antibiotic, but we give 2 or sometimes even 3, because we want to take advantage of a synergism, or we want to prevent development of resistance. So, but we never know, are they really synergistic, or they are maybe even antagonistic? Both can happen, or they can be even indifferent. So because there is no method which can allows us to see how is the efficacy of 2 or more drugs. Also here colorimetry, because it's just measuring the heat. You can see if, let's say, the inhibition of heat production is just a sum of the 2 drugs, or is it better? So I think all this interaction of drug microbes can be better evaluated by having a sum of this, especially if you have polymicrobial infection, when it gets then very difficult to interpret.
Of course. So this is one of the clear advantages there in polymicrobials. We discussed a little bit earlier how polymicrobials are tricky. They're kind of difficult, but this is actually a very strong case for using biocalorimetry to look at polymicrobial infections.
Then if I can say, this calorimetry is actually a very stupid, let's say, method, because it cannot do anything else, just measuring the heat. But exactly this is the main advantage, because at the end you want to know, are they alive or are they dead? Is their drug working or not? And you don't need to know the gene or all these things from the clinical perspective. Of course, for a researcher, he would like to know much more. But from the clinical perspective, you want to give the patient something which kills the microbe, which is invading your body. And for that, you need to know what is working and what not.
That's a very good point. And also the researchers who do want to know, they like the technique because it also doesn't affect the sample in any way. There's no probes, there's no labels, there's no, you can take the sample out afterwards and you can do all of the genomics, the transcriptomics, all of the sequencing you want, because there's no effect on the sample. You can use biopsies if you want, so it can be extremely close to the patient environment. We have the clarification now from Christina, Andrej. So are there any substances that can affect the method? So I guess, if you have your sample in there, you're looking at heat, is there anything that maybe is included in your sample from your clinical sample that would actually stop you seeing the heat being produced?
Excellent question. So I'm really happy today because all these questions are highly relevant. Indeed, in blood, I mentioned, I think the second application was blood. In blood, you have granulocytes. And as I show in the movie, the granulocytes are eating bacteria. So this phagocytosis makes them not free, but they are intracellularly. And therefore, they cannot divide and they cannot produce any heat, despite they are maybe still alive there. So the protocol I mentioned shortly was with detergents to kill the leukocytes, to free the bacteria, and therefore we can detect them. So they are, by taking clinical samples, blood, but maybe also tissue or other, that they prevent bacteria from growing. And therefore, I'm sure we can do a lot in the pre-processing phase, so that we can further optimize colorimetry to really detect even those bacteria which are maybe hidden in the biofilm I mentioned by sonication, but in blood maybe by lysing the granulocytes. But we should not lyse the bacteria. That was the challenge, because you should kill only your immune cells, not the bacterial cells. So it's in the first tests, we used 2 strong lysing agents, and all our probes were false negative. So yeah, we should develop also this.
I see. Great. Christina, please let me know if that answers your question. We have another question here. Does a lack of gas exchange within the system affect biological activity of the sample?
A very good question also from Nick. How to influence the bacterial growth also by providing them the best atmosphere. Of course, for anaerobes is no oxygen. But we saw that if we add some oxygen, the aerobic bacteria will grow much better. So maybe we can have also this test kit, which is has a lot of nutrients, of course, that we know in the media, but also have a lot of gas which is needed for growth, and this is typically oxygen. So probably oxygen is like Viagra for bacteria, for aerobic bacteria, and maybe we can shorten the metabolic detection rate then up to minutes instead of hours. And we should look into this gas exchange. We know if these ampules are sealed and the oxygen is used, then the growth will also stop and even go down. So we come to the steady state and then even into dying state. Therefore, the oxygen or the gas should be also optimized, not only the nutrient itself.
Great. Thank you very much. If there's no questions, I'd just like to kind of finish off, Andrej, on, I mean, all of the things you've meant. So we basically talked about your kind of beginning, you kind of finding this technique and applying it to the clinic 20 years ago. Very difficult. No one else could do it, but you found it. You thought it was interesting. You pioneered the clinical use. Now we've talked about a future that with you and at SimCell we're trying to develop, where we have these very, very simple standards, kits, libraries of traces, where it's kind of, you know, just plug and play, kind of, you know, as ubiquitous as mass spec in the clinical labs. But here we are today, somewhere in between. And so what would you say now to clinicians where we're not quite there with the plug and play yet? We're pushing, we're going to get there. But what do you say to clinicians to try and get them to try biocalorimetry or to use this technique in the clinic? What is it that you think would really persuade them and push them into it?
I hope that this webinar today also stimulates other to think about other applications. I showed some examples, but there are many other things which we didn't think about now. Just knowing that we have instruments, and as I heard, SimCell is bringing up instruments which are much more sensitive, up to 50 times more sensitive, so we can think about new applications. At the same time, and stimulate, of course, research. At the same time, we should also know the limits of these methods. So at the end, it will be probably a combination that colorimetry will be part of the clinical routine, but not alone. We will use it together, for example, with biomarkers, with molecular methods, maybe still with cultures, and slowly see where is it. So maybe there will be first a biomarker test, and if this is positive, then we go to colorimetry, and then maybe some additional tests if there is identification problem. But I can only encourage to do tests with the instruments which are now much better than the ones we had before.
Perfect. What a fantastic note to end on. And I want to finish by thanking everyone who attended, asked questions. Thank you so much. But a special thanks to you, Andrej, for taking the time to really explain this journey to us and to really kind of give us a window into the benefits of biocalorimetry for the future of clinical microbiology. It's been fascinating. And I really, really look forward to seeing what's coming. So thank you very, very much indeed.
Thank you as well, and goodbye to everybody.
Goodbye.